All right, so we are going to apply the laws of physics to the life sciences. Okay? So to understand how physics sheds light on biological processes. Okay? So for instance, here's a simple thing. You guys know the energy conservation principle? What is, anybody know what the law of conservation of energy means? Yes, no, no? Okay, it just states that you cannot create or destroy energy. So, if, okay, in this room, let's say there's so much energy, and if it's not leaking out or coming in, you can come 5 billion years later, energy. That's a law of conservation. Okay. So the total energy in the universe is constant, because you can't create energy or destroy energy. Okay. So let's say if you, so if a system has so much energy, energy is measured in units of joules. If you have 1,000 joules of energy, you can't do 2,000 joules of work. Do you guys understand that? So that's the energy conservation principle. So we'll look at how physics constrains biological processes or sheds light on biological processes. Okay, here is an image taken by an MRI machine. Okay, we'll learn how this image is taken. Okay, do you guys know what MRI stands for?
This is right there. Magnetic
Oh,
resonance
there you
imaging.
go. All right. All right. So, okay, MRI machines use. Okay, so let's see. What is this? Okay, what atom? The hydrogen atom. Okay. So this is a proton. This is an electron. Okay. Do you have hydrogen atoms in your body? Where?
I'm just guessing.
You guess?
Over 75% water.
There you go. Very good. Okay, so this is what we're going to talk about. Okay, we're just going to look at the physics of everything. Okay, so all we are is a bag of water. There is the hydrogen atom. Every water molecule has 2 hydrogen atoms. Okay, so and that's the proton. The proton is a positively charged particle. All the mass of the hydrogen atom is in there. This guy is 2,000 times heavier than that. Okay, and he's very tiny, but he's also spinning. So he's spinning, and he acts like a magnet. When a charged particle spins, he acts like a magnet. And so the magnetic resonance imaging uses the spin of that proton to get an image, and we'll talk about it later. Okay. All right. So. So here's a dialysis machine. What does a dialysis machine do? I'm sorry? Yeah. No.
Takes the sugar out of your blood.
No, sugar is good stuff.
It acts like a kidney on the outside.
Yeah, it acts like a kidney. Okay, so your kidney is not functioning, and so until you get somebody donates a kidney to you, you gotta have you gotta go through a dialysis machine. Okay, here's what it does. We'll learn about diffusion. Okay, so okay, let me show you a little. Okay, what's going on? Okay, so we'll talk about this more. I just want to introduce this to you. This is very important. Anytime in your body, anytime molecules have to be transported over the distance of a cell, this is what happens. So when oxygen is transported from your alveoli into your blood, this is what happens. Okay, so let me show you a picture. Okay. So you breathe in air, and there are these tiny sacs, the alveoli, and oxygen from the alveoli gets into the blood. Okay. Why does oxygen from the alveoli move into the blood?
Because there is none in the blood currently.
I'm sorry?
Because there was none in the blood.
Okay, so that is diffusion. Wherever the oxygen molecule concentration is higher, it will diffuse toward the lower concentration. Okay. So, if the concentration of oxygen in the blood were equal to the concentration of oxygen in the alveoli, oxygen wouldn't move in there. Okay? So when you breathe in, when the venous blood comes to the alveoli, this has less concentration of oxygen, and oxygen diffuses here. Okay. What else is happening? There's two processes that happen. The carbon dioxide concentration here is higher, and it's higher than in the air, and it diffuses there. Okay? All right, so when molecules have to be moved small distances, over like a few cells and stuff like that, diffusion is the process that's happening. So it's happening all over your body. Okay? Why do you need oxygen, by the way? Why do you need oxygen? Yeah, you're breathing to get oxygen. Why do you need oxygen?
The brain needs oxygen, I know that much.
Everything in your body needs oxygen. You need oxygen to release the energy from ATP molecules. You have these batteries, energy molecules. To release the energy from these molecules, you have to oxidize them. Okay? So you need oxygen all the time. Okay. So let's look at diffusion. Okay. So in this simulation, you'll see this. So here are these blue molecules, could be whatever. Okay. And here's red molecules. See, these molecules are higher concentration here. These molecules are higher concentration here. And in your body, they will diffuse through a membrane everywhere. Okay. These membranes are partially permeable. What does that mean, partially permeable? I'm sorry? Yeah, certain things can come through and other things can't. Okay? So they're called semi-permeable membranes. Okay. So now here, for our sake, we'll just remove them. Okay, well, actually we made it semi-permeable. See the holes. And you see these molecules diffuse through here, and these molecules will diffuse over here. Okay, so at cellular level, this is how molecules are transported. All right, so here's my lungs. I breathe in air, molecules diffuse here. Okay. But the diffusion is a very slow process. Okay, it takes time. How do I get oxygen to this part of my body?
Goes through the blood.
It goes to the blood. So as soon as you become big, you can't transport stuff via diffusion. You have to have a circulatory system. Okay? So as soon as you become big, any animal that becomes big has to have a vascular system. Okay? All right, so now let's get back to the dialysis machine. All right. So here's your blood vessel, okay? And here's the dialysis liquid. So, and okay, so what the dialysis machine does is, okay, or what you want to do is here's this is toxic material, urea, this, that, and the other. You want to get rid of it, okay? And so you pass this, here's a semi-permeable membrane, here's the dialysis liquid. You want to get rid of this molecule. In this dialysis liquid, there's no urea, and this will diffuse across the membrane. Make this blood pass slowly enough, give it enough time, and all the toxins will be gone. Everybody understand that? All right. Here's the problem. These guys are the good molecules, glucose and stuff. You'd lose these too. You're losing the good stuff with the bad stuff. It happens in your body. But when your kidneys are functioning properly, the kidneys will spend some money, they spend some energy, and pump the good stuff back. The dialysis machine doesn't care that. So you got rid of the bad stuff. What are you going to do about the good stuff?
Gotta put it back somehow.
Yeah. Or,
Intravenous method?
or,
Diet.
no. Is there a way to prevent the good stuff from going out there?
Putting in the dialysis liquid?
So in the dialysis liquid, yeah, just put enough concentration of glucose here so it won't diffuse. Did you guys understand that? Okay, so this is our goal will be stuff like this, okay, to understand stuff like that. All right, so all living organisms are subject to the laws of physics, okay. Everything in the universe has to obey the laws of physics. Okay. So we'll look at her as a system. This system, there's no way she can break any laws of physics. Okay? All right. So laws such as energy conservation and those governing fluid flow constrain what is possible. Okay. So laws of fluid flow. I'll give you a little example. So here's a pipe. And you can see that its cross-section is changing. This is the area of cross-section, area of cross-section. By the way, if I'm used to using physics language, and so if there's some term that's confusing, just stop me. Okay? A1 is the area, V1 is the speed at this location, A2 is the area, V2 is the speed at this location. Okay, so let's say we have some kind of fluid flowing, it could be blood, water, whatever. The flow rate That's how many gallons of fluid are going per minute or whatever, okay, however you want to measure, liters per second, is A1 V1. The area of cross section times the speed of the fluid. Okay, so here's this pipe. This pipe has no leaks. 5 gallons a minute are flowing in. That's it. How many gallons a minute are flowing out? I'm sorry? 5 gallons a minute. Again, there's no leaks. Okay. So let's write this formula down. So A1 V1 must equal A2 V2. Right? All right, but A2 is bigger than A1. So V2 must be smaller than V1. So you guys understand? As soon as the pipe got wider, the fluid slowed down. All right, why do we talk about this? Okay, so here's an example. How do your cells get molecules like oxygen, glucose, all the nutrients? Diffusion. Okay? So your blood has to bring all that stuff close to the cell, and then diffusion happens. Is diffusion a fast process? No, it's a slow process. Okay. Blood is pumped out of your heart at a foot a second. Blood is going that fast. If the blood went by that fast next to your cell, there wouldn't be any diffusion happening. So what's your goal now? You got to slow down this blood. Okay, so let's see how we'll slow down this blood. You guys understand? All right. Yeah.
Wouldn't the body design itself in such a way that where diffusion needs to happen, that the flow rate would be less than, so that it goes slower? So therefore, at certain intervals inside your body, the width of the where the substance is traveling either gets smaller or larger depending on what rate of diffusion needs to happen.
You figured out. You can design a human being. Okay? That's your homework.
Good enough.
Yeah. All right. So
No pressure.
no pressure.
I already made one human, so I'm good. So.
Yeah. Oh, no. Yeah. Well, you got to improve the design. I mean, just.
When you're sick and things happen to some people, they can't do that. So that's why you have to like figure out these type of things.
That's what I was gonna say, so naturally,
It's
sure,
supposed to be
but
that way.
when we have to do things artificially, it doesn't work the same.
All right. So let's figure it out. Okay. So you just saw that to slow a fluid down, all you have to do is increase the area. Okay. So how do we increase the area? Okay. Well, you all know. I'm sorry.
A larger baby.
No. So all you do is break it up into capillaries. Billions of capillaries. And the total area is now the sum of all these areas. Okay? So blood is coming out at 30 centimeters per second out of your heart. And here it's going at about 1 millimeter a second. That's how much you slow it down. Okay? So that's what you do. You had to break up your arteries into small capillaries where the total area is so large now that the blood is going that slow. Okay. Well, let's do a little calculation. Okay. So A1 V1. Again, you don't have to worry about any of this stuff. I just want you guys to understand this. V2. So A2, we're going to calculate how much the area increased, okay, from the arteries to the capillaries, okay. So A2 is A1 V1 over V2. A2 is V1 is 30. Again, Don't worry about any of this calculation. 30 centimeters per second. V1 is 1 millimeter. A millimeter is a tenth of a centimeter. Okay. A1. This is 300 times A1. So what we are saying is the area of all these capillaries adds up to 300 times this area. That's what we're saying. Okay? All right.
So, in a heart patient who
Mm.
has a blockage,
Mm-hmm.
does it have more to do with the rate of the flow of the blood and the oxygen to the heart and the diffusion rather than just the fact that maybe it's getting less oxygen? Is it the rate of diffusion or is it the fact that the oxygen and the blood's not getting there?
All right. So you guys have used garden hose.
Mhm.
Okay. Here's your flower, watering this flower bed, and then you want to water that flower bed. What do? You pinch it. You make this area smaller. This speed gets bigger, and the water goes further. In a heart patient, here's the problem. Here's blockage. So the area got smaller. Fluid is flowing faster here. You will learn in chapter 9, when fluid flows faster, the pressure of the fluid decreases. And one of your worries is this blood vessel might collapse. Yep. When the fluid flows faster, it can become turbulent. Okay. When turbulence happens, then you have to work harder to maintain the same flow rate. Okay. So those are the kinds of problems you'll run into. All right, so again, we just gave you an example of how you take advantage of how your body is amazingly designed, keeping the laws of physics in mind. Okay. Yeah. Physics provides tools enabling us images. So to take these images, you had to, physics principles went into this thing. Okay. This scanning electron microscope image shows salmonella bacteria invading the immune cells. Okay, so this image was taken by a scanning electron microscope.
Slide's not up.
All right, I don't know what's going on, so it happens often. So when the slide is not up, you guys have to let me know. Have you guys heard of this term, scanning electron microscope?
Yes.
Okay. Has anybody seen one?
No, they're really expensive.
Very expensive, yeah. Okay. So that's what. Do you want to know how this works? Okay. All right. Here's one thing I want you guys to remember. Everything is named most things in science are named very appropriately. Okay. So here's what you do. Let's say you want to take this picture, and it's a microscope. So a microscope takes pictures of tiny things. Okay. All right. So let's say here's the thing that you're taking a picture of. You have an electrode like this. Okay. It's a metal electrode. It's got a sharp, pointy thing.
Everything you're writing is disappearing.
Okay, so if it happens, let me know because I gotta wake it up every time it goes to sleep, it seems like.
You can change that settings in your iPad.
Okay. Oh, it's not actually sleeping. Okay, talk to me after class. Yeah. Show me after class is what I meant. All right, so you want to take a picture of this object. You're going to take this electrode and you're going to scan it like That's where scanning comes from. Yeah. What we're going to do is we're going to connect 25,000 volts, something like that. Okay. All right. So when you connect voltage like this, electrons will leak from here to there. Bigger the gap, fewer the electrons will leak. So when you're here, don't draw it too fast. So when the probe is here, the current is large. When the probe is here, the current is small. So you're scanning, and all you're doing is measuring current. When you hit a bump, the current is high. When you hit a depression or a flat spot, the current is low. So what is it that you measure? A current at every location. And then the computer turned that current information into this image. Yep.
Does the scanning electron microscope know based on the current's, does it measure the current's rate or pressure? And it can tell based upon
All right,
whatever it's hitting,
it's
what
there.
kind of material is underneath of it?
So it stops at every location and measures the current. Current is how much
And
electrons are.
based upon that current and based upon what it's touching or what it's scanning at the time, it can detect if it's like a salmonella
Oh, no, no.
bacteria
Oh,
or-
no.
No, okay.
All it's doing is measuring bumps and
Oh, okay.
depressions. Yep. You make images and then,
Again, it's not showing up.
Like a fish finder in a boat,
Hmm?
like a fish finder in a boat.
Yeah, right. So our goal in this course is to help you understand living systems and biomedical tools more deeply. All right, so here's another interesting thing. Okay, what is this guy?
It's like a blood vessel.
Yeah, it's a blood vessel, and those are your red blood cells. Okay, so that's a capillary. Okay. Now, so like I said, we are interested in knowing why everything is the way it is. Okay, so so it seems like the width of the capillary, okay, the diameter of the capillary is just letting one red blood cells to go at a time. Is that an accident? All right, it's not. So let me tell you why. So let's say if the capillary was that big and maybe two red blood cells could, maybe just a little less than two red blood cells could fit in. Yeah. To make blood flow, what do you have to do? You have to push. The pressure here has to be higher than the pressure here. Okay? And then you can push the blood here. Now, if the red blood cells were going single file, you wouldn't have to maintain as much of a pressure difference. When if blood cells get crowded like that, you have to push harder. Okay? Why do you care about that? Who's pushing? Your heart? How long is it pushing? Forever, till you're alive. Okay, so if your heart has to work just a little less, the efficiency is over your lifetime. Okay, so this width is not an accident. Okay, so you don't have to push as hard if the cells go single file. Okay, and so that's why that is like that. So, yep. All right. Yep,
Is
go
that
ahead.
why people with like Ehlers, Danlos, where they have more flexibility in their tissues and like their venous circulatory system, they tend to have shorter lifespans because the efficiency, the blood rate, and there's other factors I'm assuming obviously, but could it be that the heart has to work that much harder to push the blood through because the elasticity of their venous system?
Yeah, I mean, it's costing energy, but I don't know how it's related to their lifespan. Yep,
Okay.
yep. So the circulatory system from the heart to capillaries is governed by fluid dynamics.
Disappearing.
So neurons signal each other via electrical pulses that travel along axons. So here's a neuron, and it's connected to other neurons, and it talks to each other via electrical signals. By the way, do you guys know? So here's my view. Do you guys know what is the world's most powerful supercomputer? The brain. Okay. So as far as I'm concerned, we are 90 billion interconnected neurons. Everything else just is an appendage for that. Okay. So here's an amazing thing. This is a computer. This is about 100 watt. Its power requirements are about 100 watts. Okay, it's using the same energy at the same rate as one of those bulbs. That's a 40 watt bulb. This brain of yours is 1 billion times more powerful than that. And it runs on 20 watts. Imagine the efficiency. Okay. All right. So physics helps us understand the limits of athletic performance. Okay, so it'll tell you how high you can jump. I mean, you can't probably jump beyond this point and so on. Okay. Now, what we'll do is, you guys have heard of green fluorescent proteins? Okay, so that's how this image was taken. And now we'll see how this thing works. Okay. All right, so let's go back to, and again, so this is how the lecture is going to go. We want to understand how everything works. Okay, so just, if you don't understand something, please let me know, and I'll. We don't have a periodic table here. What is this guy? Yeah, what kind of atom? It's a helium atom. Okay. 2 protons, 2 neutrons, and 2 electrons. Okay, it's neutral. So these are the energy levels of the electron. This is the lowest possible level, and it can hold 2 electrons, and it's full. Okay? This level is empty. There's that level, that's empty, and so on. How many levels does each atom have? An infinite number. Okay, so it has an infinite number of levels. Every atom, the energy levels are different. Okay, so that's a fingerprint of an atom. For every atom, the energy levels are different. Okay? All right. Here's a photon, which is a light particle. I'm going to shine the photon on this. If it has the right energy, the atom will absorb the photon, and this electron will get excited. We'll go there. Okay, so what happened? Here was this atom. I shown a photon on it. It absorbed the photon and it got excited. Okay. Now, most of the time, this atom stays excited for only about a trillionth of a second. And what does it do?
Goes back.
It emits the photon back. Okay? So it'll emit, the electron will fall down, and it'll emit this photon. Right. Now, since every atom has unique energy levels, okay, so the photon energy, by the way, so here's the formula for the photon energy. The photon energy is H. Again, you don't have to remember any of these formulas. H is a Planck's constant, and F is the frequency of the light. Okay. Do you guys know what the frequency of the light means? Anybody? Um, okay, no, so let's do the frequency. Yeah. Okay, so light is an electromagnetic wave. So here's a beam of light traveling in this direction, and you see there's the electric wave. A light beam has an electric wave and a magnetic wave. The red component is a magnetic field, okay? So that's why it's called an electromagnetic wave, because it has both electric and magnetic field in it, okay? So let's say, so this is the electric field, this is the magnetic field, and that's the direction of travel, okay? So if a beam of light were going like this, let's say we shine a beam of light like this, and you stand there and you measure the electric field. The electric field will oscillate. So right now it's like this, then it'll go like that, but it will oscillate. The frequency of the light is how many times it oscillates per second. Okay. For this light, the electric field is oscillating about 1,000 trillion times a second. Yep. So the higher the frequency of the light wave, the greater its energy. That's what this formula is telling you. Okay. Okay. So the photon energy is that times the frequency. Okay. Now every atom emits a unique set of frequencies. So when the electron falls from here to there, it emits this photon. When it falls from here to there, it will emit a higher energy photon. When it falls from here to there, it'll emit an even higher energy photon. Okay? And so the light coming from every atom and molecule is unique. That's its fingerprint. And that's how forensic sciences identify various material and stuff. Okay, so if there's trace amounts of elements of something in a fluid, you burn the fluid and you look at the light coming from it. Okay. All right, so we want to understand how this picture is taken. Okay. There are molecules that fluoresce. Okay, and here's what this fluorescence means. Okay, so now what we'll do is, so let me show you. Okay. So, so let's say here's a cell or whatever, a body part. I take a picture with an ordinary camera. What is happening? Light is being reflected, and every part of this object is reflecting the light. So I'm getting a picture of the whole thing. But I don't want a picture of the whole thing. Let's say I want a picture of some specific parts of the object. Okay. So here's what I'm going to do. So let's say I want pictures of these molecules. I'll attach these fluorescent molecules to this molecule. Okay? And these are, so in our example, these are the green fluorescent proteins. I'll attach them, and wherever they go in the body, now I'll take pictures. I'll shine light on those green fluorescent protein, and they will emit back a light. And they'll emit back a light of lesser energy. And I'm taking a picture of just that lesser energy photon. And you see, these guys are all over there in the body. Okay? Did you guys understand that? Okay. So now let's see how that process works. Okay. So now we'll look at the energy levels of this molecule, the tag molecule. Okay. All right, so here are the energy levels of this tag molecule, the molecule that fluoresces. Okay. What you do is you shine UV light on it. So these are the lower energy levels. These are the higher energy levels. Okay, that's the atom in the low energy level, and when the electron goes there, it's in a higher energy level. Okay. So you shine UV light on it. I'm sorry?
Did you intend the photo?
Okay. You shine UV light on the tag molecule. The tag molecule absorbs the UV light. It gets excited to this level. And then it loses some of its energy via heat. Okay? That happens within a trillionth of a second. Where is that? Loses some energy via vibrational relaxation, settling into the lowest vibrational level. It goes there. This energy, okay, there's time information here. In picoseconds. Pico is 10 to the minus 12 seconds, okay? So you shine UV light on that tag molecule, it gets excited, it loses some of its energy there, and then it emits this light. This light is a lower energy light, photon. Okay, so you shine UV light on it. Most of the other parts of the body are reflecting the UV light. But that tag molecule is absorbing the UV light and then emitting a light of less energy, different color. So here's you're shining a UV light. Your camera is sensitive to the green light, lower energy light. It's not taking a picture of the UV light. And so now you're getting a picture of all the molecules that you want to take. Did you guys understand that? So that's how this picture was taken. Okay?
Contrast.
I'm sorry?
Does that have contrast? Like when they say they're taking a picture, like you have to put contrast in the picture and then take a picture?
No, you have to tag the molecule. Yeah. All right, so here are some terms. So this process is called fluorescence. Fluorescence is when you're absorbing a light of higher energy and emitting a light of lower energy. Okay? Because you lost some of the energy via vibration to heat. Okay, so that's called the Stokes shift. The emitted fluorescence is a longer wavelength. Okay, so let's make sure we understand this. This is the speed of light, c. That's frequency times wavelength. Okay, so here's the light wave. This is the wavelength. Okay. And again, exams are open book, open notes. All you have to do is just read and remember and understand.
Is that a lowercase f?
Yeah, that's a lowercase.
And then a h, is that the h?
No, that's lambda. It's a Greek letter. Lambda. Okay, that's the symbol used for wavelength. Okay, so here's the speed of light. The speed of light is constant. Okay, so if the frequency increases, the wavelength will decrease. If the frequency decreases, wavelength will increase. Yep.
Can you just say that one more time?
All right, so this is constant. This is the speed of light. Okay. This is frequency times wavelength. So if frequency increases, when does frequency increase? When the energy, so let me write the other formula. Energy is h times frequency. This is a constant. So if the frequency increases, energy of the photon increases. X-ray has higher frequency than UV light. UV light has higher frequency than infrared light. Okay, and so on. And gamma rays have higher energy than X-rays. They use gamma rays for radiation therapy and all. Okay? All right, so when the frequency increases, wavelength decreases. Okay, so here the energy has decreased, frequency has decreased, wavelength has increased. So the wavelength of this light is greater than the wavelength of that light. Okay, the emitted fluorescence is a longer wavelength and less energy, and that was what was important. Okay, so you tag these molecules, you shine UV light on it, and it's emitting a less energy photon, and so you can take images of specific things. Okay, you can put that stuff inside your body. The camera is outside, and you can take pictures. Right? It's a fast process. It happens within a billionth of a second. This photon is emitted within a billionth of a second. So you shine UV light, and this guy is coming out in a billionth of a second. Okay. So this property, the tag molecule property, is common in organic molecules. Okay. And you have green fluorescent protein was the first one discovered, but there are other colors as well. Okay. You use this technique in forensics, okay, detection of bodily fluids. In your bodily fluid, there's plenty of fluorescent molecules. So if there's a small amount of bodily fluid somewhere, all you have to do is shine UV light on it and watch for fluorescence. Okay, so you can detect that. In currency, they have fluorescent molecules. So if you have fake currency, that won't have fluorescent molecules. So if you want to see if the currency is real, you shine UV light on it and look for the fluorescence. Yeah. Okay. I want to get to slide 14. I'm going to explain this one instrument to you, and then we'll get done. Okay. I need to catch up to. Okay. What is this, guys? You guys have all seen this. Pulse oximeter. Okay. What does it measure? I'm sorry? So, okay, so the name has two things, and the pulse rate. Okay, so it measures your pulse rate and the oxygen level in your body. Okay, so let's see how this thing works. What molecule carries oxygen? The hemoglobin in the red blood cells, that's where the oxygen attaches. Okay? So here's how it works. So what you do is, okay, this thing has to be attached to a small, thin part of your body, either your face.
Disappearing again.
All right, so you have to attach it to the thin part of your body here, there, somewhere. Okay? So here's so the hemoglobin is in the red blood cells. I'm just going to draw this. It's denoted by HB. And if oxygen is attached to it, then, okay. So here's a hemoglobin molecule where oxygen is not attached to it. Here's a hemoglobin molecule where oxygen is attached to it. This guy is doing his job, this guy is not doing his job. Okay. So in that instrument, what we have is two beams of light that are that they shine from the bottom, okay? One beam is an infrared beam.
Your marks are disappearing and there's no picture.
Yep. And another is a red light. Okay? All right, so these are the two beams. They shine a red light whose wavelength is 660 nanometers and an infrared light whose wavelength is 940 nanometers. Okay? Infrared light is absorbed more by oxygenated hemoglobin. Okay? So this beam is absorbed by this guy. So by oxygen. Okay. So this guy is absorbed by that. So more oxygenated hemoglobin there are, the intensity of the infrared beam will be less. This beam is absorbed by that, unoxygenated hemoglobin. More unoxygenated hemoglobin there is, this beam's intensity would be low. Okay? So what you have is, you shine these two beams, you have detectors here, you're measuring the intensity of these two guys, and you can tell how many blood molecules, how many hemoglobin molecules have absorbed oxygen. Okay? And so that's how that works, and this is how you measure it. This is oxygenated hemoglobin, and this is the total number of hemoglobin. Okay, and what you want is this number to be between 95 and 100%. Okay, so you want at least 95% of the hemoglobin molecules to be doing their job. Okay. How do you measure the pulse rate? Any ideas on how the pulse rate might be measured? Let's go back to this diagram. I'm sorry? Very good. Did you guys all pay your fees? Okay. So every time your heart beats, more blood is flowing through, and the intensity, this intensity, whatever the intensity is changes with time. When more blood flows, the intensity decreases. When less blood is flowing, the intensity increases, intensity of light. And so from the peaks and valleys, you can get pulse rate. Okay. So that's how that instrument works. It does two jobs. It measures the oxygen levels, and it measures the pulse rate. Okay. All right, guys, that's it for today. Hopefully, okay, so here will be the goal.